Spirogyra introduction. It is one of the most common and large genus with about 300 species growing as free-floating masses in stagnant or slowly moving waters streams. Some species are attached to the substratum through a basal rhizoidal cell. The masses of plants are slimy to touch because the filaments are surrounded by pectic sheaths which when come in contact water forms mucilage, therefore it is known as water silk. The Spirogyra grows abundantly in spring. Plant body, the thallus is filamentous. The filaments are green, simple, unbranched and consist of an indefinite number of cylindrical cells placed end to end in a single row. In free-floating species, there is no distinction between the basal and distal ends, whereas in attached forms, the lowermost cell develops a rhizoidal outgrowth called holdfast which helps in attachment. Cell structure shocked face. The protoplast is bounded by plasma membrane which is surrounded by a two-layered cell wall. The outer layer is pectic and forms slimy mucilaginous sheath around the cell after dissolving in water, and the inner layer is cellulosic, i.e., made up of cellulose. A large central vacuole occupies the center of the cell. It is filled with cell sap rich in tannins. The cytoplasm is peripheral in position and is in the form of a thin layer. A single large nucleus is suspended in the center of the cell with the help of cytoplasmic strands. Many small rounded pyrenoids are present in each chloroplast. Mitochondria, endoplasmic reticulum, dietiosomes, Golgi apparatus, and other organelles are also present in the cell. The number of chromosomes vary from species to species, usually from 5 to 24. Life cycle or reproduction. The reproduction in Spirogyra is vegetative and sexual mostly. However, some species also exhibit asexual reproduction. Vegetative reproduction. The vegetative reproduction takes place by fragmentation, i.e., breakdown of filament into fragments due to mechanical stress or dissolution of septum between the cells. Asexual reproduction. The asexual reproduction, where present, is by formation of thick-walled, non-motile aplanospores, or if a gamete fails to fertilize, it develops a thick wall to become azygospore which give rise to a new filament. Sexual reproduction. In Spirogyra, the sexual reproduction takes place by special gametescaled aplanogametes and the process is aplanogamy. The motile gametes are always lacking. Aplanogamy takes place by conjugation, which may be scalariform or lateral. In each cell a single aplanogamete is produced which moves into the other cell through a conjugation tube. The species may be homophilic or heterophilic. Mechanism of conjugation. During the process, two filaments come near each other and become glued together by their mucilage. A finger-like outgrowth arises from the lateral walls of the cells called conjugation tube. The protoplast in each cell rounds off due to loss of water to form gametes. The gametes are isogametes, however one gamete is active and move from one filament to other. It is called male gamete. The other gamete is passive and remains in the cell. It is called female gamete. Therefore, Spirogyra exhibit physiological anisogamy. The male gamete moves towards the female gamete through conjugation tube and the protoplast and nuclei of both the gametes unite to form a diploid zygote which secretes a thick wall to become a zygospore. After a period of rest, the zygospore germinates into a new filament by cell division. Low nitrogen supply and high light intensity usually stimulate sexual reproduction. Types of conjugation: shocked face, lateral conjugation. This type of reproduction occurs in homophilic species. Here the aplanogametes of the adjacent cells of the same filament unite. At the septum a tube-like structure develops and through this opening the content of one cell passes into the other. Then both fuse to form a zygote. The empty cells are considered as male gametangia and the cells with zygotes are female gametangia. Very soon a thick wall is secreted around each zygote and dark-colored zygospores develop. They undergo a period of rest and germinate under favorable conditions. Scalariform conjugation. This type of reproduction is found in majority of the heterothallic species of Spirogyra. The filaments taking part in conjugation lie side by side. Very soon the outgrowths are given out from the lateral walls of the opposite cells of the filaments. The outgrowths of opposite cells touch each other. Very soon the wall of contact dissolves and a tubular passage is formed between the two opposite cells of the two filaments lying side by side. This tubular passage is called conjugation tube. Simultaneously the contents of the cells retract and aplanogametes are developed. A single aplanogamete is developed in each cell. The aplanogametes formed in the cells of one filament pass into the opposite cells of the other filament through conjugation tubes. The plasmagamy is followed by karyogamy. The transferring aplanogametes are considered to be male gametes while the receiving aplanogametes are female gametes. Just after the fusion the walls are formed around the zygote and they are called zygospores. A single zygospore develops in each cell of the female filament. The wall of the female decays and the zygospores are set free in the water. Each zygospore germinate into a new plant after a resting period.